I assume you guys already know about the PCL, but to make sure, in this quiz, we are going to cover almost everything about the PCL. Okay, let's start. Okay, one polymer chain reaction. It's a polymerase chain reaction. Okay. Move on. Okay, answer seems like straightforward, but if I ask you a question, if DNA can be amplified by chain reaction, RNA cannot be synthesized in that way? The current the system of a PCL is for DNA amplification, that's true. But in principle, it's not possible for RNA. Okay, let's think about this problem later again. Maybe almost a similar question to the previous one. Let's think. We are amplifying DNA, then maybe we use the DNA polymerase. Then if you change this DNA with the RNA polymerase, what is the product? What is the product of RNA polymerase?
RNA.
It's RNA. Okay, then what is the template of RNA polymerase?
DNA.
Should it be DNA or it can be others?
It can be both DNA and RNA.
When it is a DNA?
Transcription.
During the transcription, this is a DNA templated RNA polymerase. And you also said this can be RNA too. Then when is it the case of a RNA polymerase utilizing RNA template?
Some RNA virus.
Maybe it's an RNA virus, and they use RNA template, and then they synthesize the RNA as a product. It's also possible. And next one, reverse transcriptase. What is the template and what is the product? Soyun?
Excuse me.
Say clearly. I don't hear you clearly.
In DNA polymerase.
No, reverse transcriptase.
Is DNA is template.
DNA is a template. What is the product?
Product is RNA.
That's a transcription. That's a transcriptase.
RNA is template and product is DNA.
That is reverse transcriptase. What is a protein polymerase? Someone, what is a protein polymerase?
Your question is, what is protein polymerase?
Yes.
Protein polymerase is enzyme that
Enzyme that make up the protein. So
yes.
what is that?
Ribosome.
It's a ribosome. So rRNA or ribosome, it is protein polymerase. We don't call it protein polymerase, but if you interpret that into that meaning, then it's a ribosome. Yeah, ribosome is making protein, right? So if you think about this the process, then why only PCR can be used only for DNA polymerase? It cannot be used for other enzymes? Let's keep it as a question and let's continue. Let's see. Okay, good answer. So replication is DNA templated DNA synthesis, and that means PCR is mimicking replication. Okay, PCR is an in vitro version of replication. So if you think about the other the process, let's just say transcription. Transcription is DNA templated RNA synthesis, and template is fixed, right? Template amount is not changed. And in one reaction, only one product is generated. And to get second one, you need to do that reaction again. Template is fixed. The only product is not exponentially. It's just a step by step. One by one, it is added, right? Translation, what is the template?
mRNA.
mRNA is a template. Product is a protein. It's the same situation. Template amount is not changed. Only product is made. So you can amplify, but it is not exponential manner. For replication, one DNA make a two strand, and then both of them can be used as a template. That's why next step you can get 4 and 8 and 16. That's exponential. Reverse transcription, template is the RNA, and product is the DNA. If you don't make more RNA, then that's also it's not exponential. It's also only product is generate. One cycle, you generate just one product, okay? So all other process we utilize for fermentation and in vitro transcription or in vitro translation, we do, but amount of generation, the speed is not exponential, only replication. Product can be used as a template. That is very unique in PCR. That's why it can generate the product exponentially, very quickly. Okay, that's a unique part of a PCR. Okay, maybe the term of DXTP is not familiar to you. So if you look at that, this is a deoxy, ATP, GTP, CTP, TTP. So that kind of random number, sometimes we use X. So this is a triphosphate of building blocks. So this is one of those components. And here, if you chose this one, during those the PCR, we need to have a thermophilic enzyme, not thermophobic enzyme. Okay, thermophilic means the enzyme which can survive and also which like the high temperature. That's the thermophilic. Thermophobic may be opposite. So I tried to give some little trick here. So the others maybe now from now on, I wish you guys are familiar with this term, DXTP or just XTP. Just XTP may be for RNA building block. DXTP is for DNA building blocks. Okay. Don't know who is the cat. I see some cat
Sorry.
beside you. It's only to me.
Sorry.
Nobody can see. Dong's video, I can see one cat head.
Yes, I can
Um,
see too.
Is it real cat?
Yes, it is.
Oh, I see. I saw it's just kind of some artifact on the screen. Okay. Dong is studying together with the cat, white cat. That's okay. Wow, this is surprising. And one of you is not answering. So I get only 6 answers. Everybody's here, right? Okay, I assume that you know about the PCL. Just that, we don't have a chance to review this. That's why I tried to make the many questions about the PCL. So in PCL, there is a primer attachment in the beginning. You have a template, some length of a DNA. You wanted to make a copy. And you add the template, the primer. Primer is attached at the one side. And then DNA synthesis happening after that template, after the primer. After synthesis is finished, now you have a double-stranded DNA. That double-stranded DNA cannot be used for the next step cycle. So you need to dissociate those two. That dissociation we call DNA melting. Okay. So it's a little different the meaning of a melting from solid to liquid, but dissociation of a double strand into one strand each, you need the little high temperature. That is what we call melting. Now, the beauty of PCL is after you are done with the one cycle of reaction, and you don't need to decompose those leftover reagents. So here the leftover reagent can be XTPs, ATP, TTP, CTP, GTP. Those reagents, they can stay there. Okay, that's the beauty of PCL. Okay, let's just think about the first point. Single-stranded DNA. If your template is only single strand, now you have a primer. Primer is attached to the right position, and the product is, first step product is double-strand DNA, isn't it? Once that first step is done, then they can be dissociated, and then each strand they can be used as template, correct? So that means single-strand DNA also can be used for template. Okay. Now, second one, thermophilic enzyme is necessary. Yes, to make that melting, you need to increase the temperature, maybe up to 90 degrees, almost near to the boiling point of water. Then common enzymes or common proteins, they cannot survive. We believe that our body temperature is about 37 degrees. So we believe most of those enzymes and proteins, they are stable and best the performing near 37 degrees. Maybe a little high temperature, maybe 40 degrees, 50 degrees, 40 degrees, already we believe our body is in danger. that small difference of the temperature can make many of those proteins can be denatured. But one key component of this PCR, okay, the idea was quite simple, but the optimization is not simple. You have a template, and originally it was double-strand DNA. You need to melt by high temperature, and if you don't want to add up more enzyme every cycle, then the enzyme should survive up to 90 degrees. That's quite a difficult job to achieve, right? Maybe you have heard how those inventors solved that problem. Anybody can guess the answer? How did that inventor solve that high temperature problem, stability problem of the enzyme? We have 7 of you. At least one of you may know the answer. Don't be shy. If there is your project, how will you solve the problem? Doyun?
The scientists found that the enzyme from thermophilic germs
Okay, where is that thermophilic germs?
in volcano.
Volcano or hot spring, yes. So he went to the Yellowstone, it's a hot spring. In hot spring, it's a difficult environment for life. The life can survive, but there are bacteria. And then he went to that, the hot spring, very high temperature water, and collected the bacteria and believed that there may be some bacteria survive, then their enzyme should be very stable at high temperature. Yes, he collected the enzymes and modified, and that is the key component of this successful PCR. And that person was working for a company, and he was not very academic person, maybe working only during the 9 to 5, and then enjoy his vacation and weekend, the surfing. And when he got the Nobel Prize, some of those academic people were not happy. Hey, we are working whole life only for this science and research, but this guy is just found one small thing, and he got Nobel Prize. Is this fair? But result-wise, this PCR is one of those biggest invention in modern biology. Without this PCR, there is no sequencing. Okay, without the PCR, no analysis for DNA. So next step is, this is amplification. Now next step is the sequencing. For the sequencing, we need to have a tangible amount of samples. So this really revolutionized and open up the modern genetic engineering, the whole field. So that's why I believe this is worth getting Nobel Prize. Now, beauty of this PCR is the third one. Every cycle, you don't need to add up anything. You just mix everything together. Invisible template, and just add up enzyme and primers, and you add the XTP, and all you do is just increase temperature and melt, and then decrease the temperature and increase. And you just do this temperature cycles. Then everything is automatically can be done. Of course, it should be very well optimized, but that is the beauty of this PCR technique. Just repeat these reactions, then you can get exponentially amplified the sample. Now, last one, single primer. If you have only single primer, you have a template like this. You need to start this side and this side. Then you can make a both strand. It is a template. That means two strand is not same. Those are, one is sense strand, the other one is anti-sense strand. They are not same. That means you cannot use the same primer for both. You need to have a sense here, and then you need to primer on the anti-sense. Then they can synthesize and together. So if you use only single primer, it is the same as a transcription. Every step you can make only one copy. It does not make double copy. So then it cannot be exponential. Okay, so that is important part of the primer. So let's check whether you are clear for primer. Okay. The first red one is the definition of a primer. You need to have a leading sequence which is attached to the template, and then DNA polymerase starts to synthesize following that primer. Okay, add up more materials to the primer. That is the primer. Usually for the primer for DNA synthesis is a DNA oligomer, correct? But if you add RNA oligomer, it can also act as a primer. If you add the DXTP, then after RNA oligomer, they add DNAs, but still RNA oligomer, they can act as a primer too. Last time I told you that DNA, they can make a double strand. RNA, they can make a double strand too. RNA also can make double strand. So peptide oligomer cannot. It's a different structure. So peptide oligomer cannot act as oligomer. Now it's time to think. If your current PCR is only for DNA amplification, but if you design those RNA polymerase with a different primer, in principle, you can make RNA exponential the increase. In principle, that's also possible. Either you can use the DNA template and make RNA, or RNA template RNA synthesis. If the product should be template, then it should be RNA templated RNA synthesis, right? It's a little more unstable molecule, but in principle, if you design proper enzymes and also proper the condition, in principle, that's also possible. But RNA is more unstable than DNA. So currently, the popular one is the DNA PCR system. Okay? Maybe you randomly just picked up. Okay, this problem we manage it later. Okay. So this is the last problem. If you pass this, then you understand Fischl. Okay, very interesting. Many different opinion here. Okay, the template of a DNA can be any size. You decide from long DNA, you decide those starting point and end point by primer design. Okay, then the primer should be a specific sequence for starting point and end point. Now, total length of the target was 100 base pair. And then primer starts to recognize it from 11s. That means they give up first 10, correct? And then the other end also give up first 10. And if primer size, now primer is a part of the product or not? Here, to make it simple, I didn't give you the size of, I already gave you the size of a 10 primer. Now, primer is a part of the product, or it is not part of the product.
Not.
It is not. You have a primer, 10 more of a primer. Synthesis happens after this 10, they add up. Then if you make this DNA at the end, primer is a part of it or not? Primer is included in the product. Okay? Section clear? Not clear?
But they are not connect 12.
Primer is a DNA oligomer. Primer is, let's say, in the previous problem, primer, let's say 20 more. Here, I just said, this is conceptually 10 more of a primer. That's a DNA 10 more. And then more DNA is added to the primer to finish up those syntheses to the end of the template.
Yes.
Then primer is included in the product. So in this problem, only first 10 is removed. Last 10 is removed. Then out of 100, you should have 80. If this part is not clear, you should go back to the PCL and then figure out, okay, what is the mechanism and also how it can be decided. Okay, we cannot spend more time for this for now. So let's finish it here. If you are still not clear, then we can discuss that later again. Okay, coming back to the class. Okay, today's the main part was a PCL. Section still it's not clear.
I understand it.
Okay. Any other question for PCL? If not, let's come back to that other question I gave you. To make a primer, if you make a primer too short, then they can bind to many different sites in DNA, right? Our DNA total length is 3 billion base pair. We have another copy, two copies, almost the same, but X and Y is different. So if you make the primer too short, then they have a multiple chance to bind to different sites, right? Then to get specificity, I wanted to select only this sequence. Then we need to have reasonably long primer. If not, they have multiple chance. Randomly, they can bind to different sequences. Then how many sequence, how many lengths, how many base pairs is the minimum length to avoid that random recognition? That's the question. And to get the number, you can try to count how many different combinations you can make. So let's try. If it's 5 more, how many combinations do you make? Okay, if it's a dimer, how many combinations do you make? DNA dimer.
16.
It's the 16, right? 4 by 4. If it's 3, 4 by 4 by 4, 2, 5, 6, right? Then if it's a 5,
1,000.
it's about 1,000. 4 to the 5, 4 to the 5 is the same as a 2 to the 10, right? Correct? So 2 to the 10, 1024, correct? So 2 to the 10, we can convert into 1,000. It's an easy number to convert, right? So then, if it's 5 more, about 1,000. If it's a 10 more, how many?
Million.
Million. Very good. 15 more.
Billion.
It's a billion. If it is a 15 more, the random, the kind of the species is almost a billion. Billion different kind of thing can be generated, right? Then our genome length is about 3 billion. Now, if you have a billion different choices, they bind to 3 billion randomly. The chance is maybe 3 binding site, correct? If it is totally random. If our genome sequence is totally random, if you generate 1 billion of different sequences, the random binding chance for same site is 3 copies, correct? Now, if you increase further, maybe 16 more. Now we have 4 billion different combinations. Then statistically, there's only one binding site approximately. 4 billion sequences we have, and on our genome sequence size is the 3 billion. 4 billion to 3 billion, it's almost a 1-to-1, isn't it? So to increase that specificity, we can add a little more. So usually, practically, when we use the PCR primer, usually we use about 18 to 22 more, about 20 more of the oligomer. Then we believe that we can avoid those random recognition to the genome site. Does it make sense? Okay, so this is up to the PCR. So with this, I would count that we understood those PCR. If some of the part is not clear, then with these problems in mind, go back to the video clips, and then try to look at again, and then figure out which part you couldn't understand exactly. Okay. Now, DNA amplification utilizing this PCR, amazing technique. Even though there is a one copy, just a single copy of a template, in principle, we can make the huge numbers of the DNA by this amplification technique. And this can open up the new era of the genetic engineering, maybe 30 years, 40 years of history, but that made almost everything in the modern biology. So this exponential amplification is a really important technique. Now, once this is done, next step is we want to know those sequence, okay? So today's your presentation will be mainly focused on this sequencing. So Yongun will start with the one technique, Maxam-Gilbert technique, and then Taekjun will attack with the Sanger sequencing, and they will argue together which method is better. And then after the 30-40 years, and new technique, so-called next generation, our generation, okay? Next generation sequencing is introduced. And if you have a time, sorry, the Dongwon's topic is the nanopore, the technique. I'm not sure whether we have enough time or not. If we don't have enough time, then Dongwon's the presentation will be combined with the Juan's, so for some time later. Okay, so then let's try. Let's start. Youngwon, would you share your file and start?
Yes.
Okay.
Hello, my name is Choi Young-won, and today I will talk about the Maxam-Gilbert method. Contents are like this. Firstly, DNA preparation should proceed. DNA should be separated for this sequencing, and it is separated by heating. And then separated DNA, so to speak, one strand is distributed equally to 4 samples, and 4 samples are labeled radioactively by 32P, and 32P takes on one site of the phosphate group of the 5' and the deoxyribose. And then chemical treatment should proceed. First sample is treated with formic acid, and formic acid breaks the link between the furans and deoxyribose. Second sample is treated with dimethyl sulfate, and dimethyl sulfate methylated guanines, and methylated guanines are removed from the strand. Third sample is treated with hydrogen, and hydrogen hydrolyzed pyrimidines, and hydrolyzed pyrimidines are removed from the strand. Fourth sample is treated with hydrogen and sodium chloride, and hydrogen makes a product by the reaction with sodium chloride, and that product only attacks cytosine, and only cytosine is removed from the strand. And finally, hot piperidine is treated to all samples, and hot piperidine acts as a seizure, which acts on the site where there is no base. So there can be various lengths of strands in all 4 samples. Now, samples are all completed and they should go on electrophoresis. 4 samples are loaded on the jar, and as you all know, these samples move to the right, and shorter ones migrate faster than longer ones. And to analyze autoradiobiography is used. X-ray film, gel is exposed to X-ray film overnight, and then on the X-ray film there are black bands of the DNA. So to speak, it visualizes the DNA band, and bases can be inferred from by this. As you see the right picture, there are 4 red circles, and let's see from the under. For the first circle, there are two bands at the same height for T plus C and C, and this means that the base that is on this side is cytosine, and for the second circle, there is only one black band on the T plus G, and this means that the base on this side is T. And for the A plus G and G is same means that the analyzing method is same, and because it's symmetrical, by this way, sequencing is possible. Thank you for listening to my presentation.
Okay, before we get any question, let's hear the Taekjun's presentation, and then let's compare. Taekjun, would you go?
Okay, I will explain about Sanger sequencing. Sanger sequencing used chain termination principle. So this picture shows the polymerization of nucleotides. We saw this picture in Monday. So 3' hydroxyl group in 3' and triphosphate group in 5' carbon binds together and makes phosphodiester bond. And if the nucleotide don't have hydroxyl group in 3', the polymerization can't bind. And that molecule is this Dideoxynucleotide. And Sanger sequencing used these molecules to terminate the process and can sequencing. Sanger sequencing developed by Frederick Sanger in 1977, and this method used chain terminating deoxynucleotides. And this sequencing makes new strand of DNA. So it needs DNA template and DNA primer and DNA polymerase and DNTPs and DDNTPs. The classical method of Sanger sequencing, the DNA sample is divided into 4 separate reactions like this, and each sample have DNA template and DNA primer and DNA polymerase and DNTPs or same, but each sample have only one kind of deoxynucleotide. The first one have only DDHPs and second one DDTPs and so on. And after process of DNA polymerase, then it's through into gel electrophoresis. And as you see this picture, the direction of electrophoresis is up to down, and the short DNA fragments are faster than longer one. So the short one go posing bottom. So read sequence from bottom to top, and we can sequence the DNA. And another process used I-termination sequencing. In this case, they use fluorescent molecules like this, and they emit different lights. So they, in this process, we use 4 kinds of DDNPPs and 4 kinds of fluorescent molecules. So in this case, we don't need 4 separate samples. We just need just need only one sample. So we use all TDNTP in one sample, and after the process was end, we use gel electrophoresis, same as the previous method, and then the laser pack the molecule, then the fluorescent molecule emit a different light. So like DDATP emits green light and DDCTP emits blue light, like this. So computer check the color of light, and then we can know the sequence about DNA by light. Thank you for listening.
Okay, very nice presentations, both of you. Could you take out your file? Okay. Now, to me, it's a very clear and good presentation. I wish it's the same for all of you. If anything in these two methods is not clear, you give a question. If you don't give a question, I will give you a question.
Actually,
okay, yeah, go ahead, Sejin.
Yeah, I don't understand how to analyze the radioactive label in Maxam-Gilbert sequencing.
Okay, Maxam-Gilbert methods, they use the radioactivity, and in Sanger method, later part, some of those fluorescence molecule was introduced, but those kind of combinations can be shuffled in any combination. But let's try to answer. Maxam-Gilbert method radioactivity. Sejin, what is your question again?
I don't know how to analyze the radioactive pairing. What is A, what is G, what is...
Okay, so that may be the key part of a Maxam-Gilbert method. Youngwon, would you explain again what makes those A, what makes the A plus something?
Yeah. First of all, the radioactivity is used for the X-ray film. By the radioactive labeling, there can be black bands on the X-ray film. And the second question is about the A plus G, and so I explain in more details. Can I 화면 공유?
You can share that.
Yeah.
Can I share my screen?
네.
Okay, first part, I think that is not the main point of Sejin's question, I think. Radioactivity can be transferred that energy to any kind of a film. So it can be white or the black. So radioactivity can be converted into visible signal. So that may not be your question, I guess. Okay. So then Youngwon is going to explain what makes this A plus G or the G kind of things. Go ahead.
The shorter strands migrate faster than longer ones, and so the shorter ones means that the start position of the strand, and the same band on the two sides means that the A plus G, T plus C, and C means that the bases are removed. The basis that is.
Okay, let me help you a little. So in both cases, Sanger method and also Maxam-Gilbert method, they have this kind of, you don't see my other cursor. You use some fragmented sample from original DNA sequence, correct? So then the key part is, how do you generate that kind of a fragment? That is the key part. Okay, Youngwon, would you take down your other slide? Let me share my slide. Okay, the key question is, you see the Youngwon's slide, right? Everybody, now you see the Youngwon's slide, correct? You see my,
Yes.
okay. This is the original sequence, okay? We want to know those sequence, the order of this. Now, both method, Sanger method and also the Maxson-Gilbert method, we generate some kind of a fragment and then run this kind of electrophoresis, okay? If it is 4 different sample, then we may want to have, ideal case is maybe we want to have A, G, T, C. Now this 4 means which side we can cleave chemically. Maxson-Gilbert, they are Harvard chemists, and they look at those DNA structure, and very smart chemists. By looking at the chemical structure, they tried many chemical the reagent and then found which chemical selectively cleave different positions. They could not get all selective ones, all ATCG, but one reagent they can cleave only G site. One reagent they can cleave both A and G. The other reagent only cleave maybe C, and then the other reagent cleave both T plus C, T and C together. And then they added randomly the amount. So that's why the random cleavage happens. Not random, but specific site. But if there's a multiple site, some position here, some position here, some position here, they generate all fragments. Visualization, they used radioactivity. Then, depending on where they added in the front side. Then you cleave here, then you have 2 fragments, one fragment here and the other fragment. But the visible one is only this side because your reporter is here. The other side, you don't see them, right? So then the longer one, you can see this one, shorter one here, next one here, if it is cleaved after T. Then you can, after C, let's say C here, 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 here. If that reagent added here, then you see 4 different bands. And you can decide which is the side by this length. Same thing you repeat for G. Ideal case, it could be done for A, that would be simpler, then you don't see this band. But unfortunately, they could not find all 4 selective, but the selectivity is given only to G, and this one is A side and G side together. Okay, so that's why if you see both of them, this should be G. If you see only depend only in A plus G, then this should be A. So utilizing the chemical selective cleavage, they generate this kind of a fragment, and then they made this 4 different gel running, and then they counted, they started to read this sequence one by one to rebuild these sequences. So in comparison, Sanger method, what is the difference? Taekchun, would you explain?
Sanger method makes new DNA strand. They
and
don't.
How did you make that fragment?
They use DNTP to terminate the DNA polymerase process.
So the biggest difference between Maxson-Gilbert and Sanger is Sanger method is a resynthesized those fragment, but they added dideoxy terminator, so they could not grow further. So they stop here, here, here. If you control those amount, then you can control those the relative amount of this band. That is their method. Which method is more similar to PCR?
Sanger.
It's a Sanger method, right? PCR is almost similar to Sanger method's amplification application, but reagent and also the chemicals, everything is very similar. Only difference is in Sanger method, you don't need this repeat. Just a one-step reaction is fine. But in PCR, utilizing this biological method and utilizing terminator, and then it's repeated. So now, whether this is a radioactivity or fluorescence, that is not the difference. That means even in this Maxson-Gilbert method, they could add different fluorescence material on top. That's also possible. And if you use only one fluorescence molecule, then you need to run 4 gels. It's the same as a radioactivity. Radioactivity, you cannot discriminate the different radioactivity. Radioactivity is just radioactivity. So you need to run 4 gels. If you use only one fluorescence color, then you need to run 4 gels. But if you use 4 different color of fluorescence, then even Maxam-Gilbert method, you can run in 1 gel. And you just count, you just discriminate those different color by this different color coding too. That's also possible. Now, in Takjun's the presentation, he used terminator fluorescence building block. That's one possibility. But you can also do primer side here. You can add fluorescence here too. Then your reading is from here, only upper side. If you use a terminator side, then your reading is from bottom side, and then read up. Okay. So also method, you can mix up either fluorescence method or radioactivity method. That is not the key difference. Key difference is one method is chemical cleavage, selective cleavage. The other method is a biological method, resynthesis, but stopping in the middle. Okay, those two are key difference. So these 3 people, they got the Nobel Prize together, 1980. Both method was the awarded the same Nobel Prize they shared. And which method is superior, do you feel? Which method is better?
Sanger.
Why?
Because Maxam-Gilbert process needs a lot of chemical that very toxic, and it is very complex to have complex process that cut the DNA fragment one by one.
Okay, Youngwon?
I agree for the complexity and toxicity, but in Sanger method, primer is needed, and I know as that because of the primer part, and we can not actually analyze the primer part sequence, but so in that point, accuracy may be more higher than Sanger method.
Why accuracy is higher in Sanger method?
Because I mean accuracy is higher for the primer part cannot be sequenced, as I know.
Okay, in Sanger method, you need to use a primer. Primer, the meaning of you use the primer means at least you knew partial sequence of your sample, correct? If you did not know this part of sequence, then you cannot design your primer, right? That means in Sanger method, actually you need some pre-existing information about your target. If not, you cannot start. And the second point is this much of the primer, if you use, then this much of information you already knew, or at least from sequencing, you cannot get those informations. So conceptually saying that Maxam-Gilbert, even though you do not know any information about your target gene, still you can do it. You said it's more complicated, but in a sense, you just divide your sample into 4 and just add 4 different reagents just once, and then run the gel, each of them. Well, conceptually, it may be more, it may be simpler. In Sanger method, you need to have your template, you need to design your primer, and you need to add the enzyme and let them make those the product, and then they let them stop in the middle of somewhere. Stepwise, it is more complicated, isn't it? Toxic chemical, well, that's a little different kind of issues here. So byproduct of a biological process of the chemical process, both of them has a problem. Except that kind of the issues, conceptually, Maxam-Gilbert was much more straightforward and easier and understandable. So that's why when they developed these two techniques, Maxam-Gilbert was much more popular in the beginning. But later, now we need to handle very long sample and repeated process like a PCR. Then more and more, Sanger method, in the beginning, the conceptually is more complicated. You need to combine different components. But to handle large numbers of samples and very long sample, this is similar to PCR. That's why breaking up to those the band and analyzing by chemically versus biological production, biological method was getting more and more popular. Now, currently, as we can see from PCR, this Sanger method survived, and Maxam-Gilbert is disappeared in the history. Okay. Now, most of those DNA sequencing is utilizing this Sanger method. Now we will hear one more talk by someone for NGS. Even much higher speed and also much more samples can be handled using this concept. And let's hear about that part. Someone, would you start? So assuming that we understood those Sanger method, so you just focus on those NGS part. What makes NGS?
Yes. Can you see my screen?
Yes.
Okay. Today I will presentation about NGS, next generation sequencing.
Would you speak a little loudly? So your voice is a little small.
Yes, this is the table of contents. Differences between Sanger sequencing and NGS, and pros and cons of NGS and its application. NGS is high-speed sequencing method, and it can analyze massive DNA sequences in parallel. In Sanger sequencing, as Taek-Jun explains, DDNTPs are attached in strand, and we can detect colors and lengths, so we can obtain the DNA sequences. Otherwise, NGS have some different mechanisms. There are variable mechanisms for sequencing or applications. Simply, the point is, in NGS, DNA fragments are sequenced simultaneously in parallel. So also we can obtain DNA sequences in short time. After we sequenced DNA fragments, and by aligning overlapping sequences, we can obtain whole of the sequences. For this simultaneous sequencing, firstly, we need to library preparation, which fragment DNA to many fragments, and ligation adapters for sequencing. Pros of NGS is reduced cost and time, and it can it can sequence massive amount of DNA sequences in comparison single sequences, but its cost it is hard to grasp sequencing quality. And because of this complexity of analysis, we need more analysis algorithm. So we can analyze a massive amount of DNA sequence at first, and after that, we can test the accuracy with Sanger sequencing. NGS can apply it to many parts, but the typical one is gene therapy. The cause of cancer is various, and mutation of DNA is one of them. Analysis of whole genome sequences, and we can detect the mutation of DNA, and we will treat cancer earlier. Thank you for listening.
Okay, thank you, Seung-Won. I'm not sure whether the key idea everybody got. Any question for NGS? If anything unclear, then you can tell.
I have a question.
Go ahead.
To start the process, we need adapter. Adapter is right.
Yeah, adapter.
Yes.
Okay.
I want to know how we can attach the adapter to the ends of the DNA fragment on each DNA fragment.
Can you answer, someone? Okay, before we go to the technical part, let's check the basic one first. What is the critically different of this NGS from previous sequencing data?
Utilize computer calculation.
Just the computer analysis, is that the only one you think? We need a high power of the computing? Yes, we need that because the data amount is huge, but more hardware and also experimental part is critically different. So let me summarize the conventional method. Let's just say Sanger method is you resynthesize those fragments of DNA and you introduce a fluorescence molecule here and there. It can be fluorescence or it can be the ion current, different method, but it doesn't matter. It's a generating fragment, and then you isolate those fragments by gel running or electrophoresis. Electrophoresis is a gel in the small tubes. Anyway, you can see those different size of a DNA, and by reading different method, DNA or the radioactivity, the fluorescence or radioactivity or ion current, different method, but those are just the conceptual difference. But key difference of NGS is something else. Conventional method, you can do only one sample at a time, right? Biggest difference in NGS, you can handle, someone said massive samples. Here the massive means, does not mean 10 or 100. That means thousands, millions, or maybe billion, I'm not sure, but maybe in principle up to billions of different samples you can handle at the same time. Computational thing is afterwards. When you get those data, then you need high computation, correct? But first part of NGS is not computational part. That's hardware part. How do you manage that millions or minimum millions or billions of different samples at the same time. That is the key part. If you did not get that one, then you did not understand NGS properly. Anybody can try to answer that? No? Okay, then let me help you. Someone made the two versions of this presentation material. Let me combine those two. Okay, first one was adapter issues. You have these samples. It can be mixtures of a different DNA, or it can be maybe many, maybe long DNA sequence you wanted to know, and then you can break them up and then make a and then generate many fragments like this. This can be mixtures of a partial fragment of this long DNA. And then you add this kind of adapter in both sides. Let's just say your target is you want to sequence them all. If this is 3 billion of human genome, it's too big, you need a fragment. If this is mixtures of different peoples, then still you have many, many samples like this. This is your target. And you add these adapters. And then key part is utilizing this adapter. Now this is the adapter part. Okay? This is your different samples. Now this adapter is added here. These adapters are kind of primer for PCR. Okay. Now on the bottom, you can have, on the bottom, you have both sides of primers. You need to have two primers to make PCR, right? That's why if you have this primer on the bottom randomly, and then one sample attached to one side, the other sample attached to the other side. So these three are different samples. And then utilizing these two ends, this is one primer and the other side, this is another primer. And then they are randomly distributed two primers. They make this band, and then they synthesize DNA, and then open up, and one is the stand up, and then regenerate this kind of a process. This is kind of a local PCR, and they make thousands of copies. This is one clone, this is another clone, this is a third clone. Then this kind of clones can be spread out in whole chip. They can be millions of different clones like that. Millions or maybe 10 millions or up to billions of different the sequences, different colonies and clones. And then they can be synthesized from here to up to here. And by reading them, similar to a Sanger method, and then they read this sequence, synthesis or different method, and then they analyze this sequence together. Here, all the sequences are either sense sequence or anti-sense sequence. You can read one by one, or maybe both of them together, and then analyze utilizing computational power, analyzing all the sequences here and all the sequences here. They are same sequence, that's why it gives you one sequence or two sequences, sense and anti-sense. Same thing here. So all those sequences you can align, and then those aligned sequences can be analyzed like this. Originally, let's say this is a long sequence, and then some fragment, there may be some overlapping part. It is randomly fragmented. Then, utilizing this overlapping, you can reconstruct a whole gene the sequence or make the different sequences all together. So to summarize, this NGS method is combined with these two techniques, combined with this highly massive, millions to billions of different samples amplification at the same time. That is the key part of a massive sample handling. And then computational analysis. That's why it is super fast. Previous method, there was a gene test, whether he is my father or not. You have his sample and my sample, and then test the two samples side by side and compare. You just did only two samples, right? If you wanted to find the criminal, then you have one standard sample, and then maybe compare with 100 different samples, but tested one by one. But this NGS samples, you have one mixture of a sample which has millions of different sequences. You can do it in one experiment. Okay, that speed is comparing to previous one. It's a really different generation. That's why we call this technique as next generation sequencing. There are different versions of amplification. Sometimes you use this kind of spots on the chip. That's one way. Or you can use a droplet, small droplets, and then each droplet carries one primer, and then amplify drop by drop, and millions of drops can be analyzed. That's another technique. And reading-wise, using fluorescence or ion current, different kind of thing is necessary. So anyway, NGS, to summarize, you need to have a massive parallel amplification, and then sequencing them one by one, but at the same time. Then you need to have a very high power calculation later to handle that millions of different sequences. Okay, so this is the best description I can deliver you. Utilizing this NGS, now we are in a different, the new, the period of handling many different kind of the tissues or samples, or sometimes maybe bacteria living in our body together. Maybe we can sequence them all together at the same time, and then analyze them by high power computer later. Okay. So unfortunately, the Dongwo, you don't have enough time to present your the material today, but your material can be used together with one later. So sometime several weeks later, I will call you again. Okay. Okay, let's come back to my slide. Many of you may be curious about the midterm exam. Midterm exam, yes, we will have a midterm exam. But unfortunately, we cannot do the face-to-face the approach. Here's my sorry. Okay. So next week, midterm exam, we are going to have the online midterm exam. So you need to present the same as the regular class. You come to here, then I will guide you how to take those exams. The exam date, it will be May 5 is Children's Day, so it will be May 7. Now next week, the next week, midterm exam week, we'll have the exam. Yeah, it's a 7th, Thursday, 2 p.m. So hopefully you don't have any conflict for the time. And next week we'll have a class. And next week the class will be the Tuesday, the 2 p.m. Oops, the topic is different. Next week is the next. What is it next week? Is epigenetics next week. And deadline for quiz is Tuesday, the same way. Now next week, Thursday is also holiday, so we will not have class. And quiz, so far, I tried to make you some quiz to answer to my questions. So seems like I'm limiting your creativity. So this time I will just give you each section. You give the questions. After you study your material and you give the question, then we'll think about that problem, rather than I give you the specific problem and you answer for those other questions. You will see what kind of format. Now, assignment 7, good for you. Next week, I will not give you assignment. No assignment next week. But this book, original plan was from this book, you choose one topic and then make one presentation from this book. But this week, no presentation material, but I recommend you read this book. Okay, that's on your own. Next week topic is epigenetics, And this book is a very well written epigenetics. In Korean, that's a Husong Yujanap. So that's a strongly recommended. You read this book, okay, by yourself. Okay, so this is at the end of the today's lecture. Any question for midterm exam or next week class? Next week we will have a class only for Tuesday, no class on Thursday. And then the next week, Thursday, we will have a midterm exam. You come together to this class, and I will give you a guide how to take the class. Okay. Okay. If you don't have more questions, then we finish today's class here. Okay, thank you guys.
You.
Thank you, Professor.
Thank you, Professor.
Thank you, Professor.